so much for having me here today. I'm really excited to share some interesting findings from some recent work that I wrapped up for my master's as I move into my PhD now at Penn State. This was a collaborative effort with a bunch of really helpful and important folks that you can see up here on the screen. So without further ado, I'm going to go ahead and jump in with some motivations behind this work. So as we all know, the long era of fire exclusion in this region and across much of the US has resulted in environmental degradation. We've seen a lot of mesification effects here in the mid-Atlantic and eastern US. That's resulted in loss of biodiversity, potentially increasing abundance of ticks, as we'll get into, among other issues. And in recent decades, prescribed fire, as we all know, has become more frequently used as a forest stewardship and management method to try and combat and reverse some of these forest declines that we've seen. However, despite the fact that we are putting more fire on the ground across the board, mid-Atlantic fire effects are largely understudied compared to our Western US counterparts, where mega fires seem to dominate much of the narrative. So we were interested in first exploring how prescribed fire is impacting these forests, more specifically thinking about potential links between tick habitat suitability and how prescribed fire may help us reverse some mesification trends related to this. Additionally, there's been some really interesting work completed recently exploring public and manager perceptions of prescribed fire here in PA and New Jersey with some really cool local folks also seen here, some of which are here. And what I want to draw attention to from this study was an unexpected and really important finding, which was that among managers and recreational forest users across PA and New Jersey, a top perceived benefit of prescribed fire here was tick reduction. Although when you turn to the literature and existing studies, that relationship hasn't quite been confirmed for this region. And so this further motivated our research questions. If you're unfamiliar, there are 3 primary tick species here in the region, but for the sake of this research, the black-legged tick is the primary one of focus. This is the tick that carries Lyme disease, so it's particularly important. And I also want to draw attention to the fact that the summer and spring months are those when humans are most likely to encounter a tick, therefore we're most likely to contract tick-borne disease, as nymphs and adult ticks have already had a blood meal from an animal where they've been able to potentially come into contact with different pathogens. And additionally, through time and space over the last few decades, we've seen quite a drastic increase in the species distribution and range in which we're finding black-legged ticks. So this map on the left shows in red areas where black-legged ticks were established in 1996, as compared to 2020 on the right. You can see quite a drastic growth in red areas, denoting a large expansion of suitable habitat where black-legged ticks are being found. And this is particularly important to consider in relation to increasing prevalence of Lyme disease as well. I'm sure many of you have at some point found a black-legged tick on yourself. I know I did when I was doing field work for this. And so understanding different drivers and mechanisms behind where we're finding these insects and not is really important for human health, especially here in Pennsylvania, as I wouldn't necessarily be proud of this, but we are one of the leading states year to year in terms of Lyme disease prevalence. And with these increasing ranges of land where ticks are residing, as well as increasing disease prevalence, there have been several or dozens of studies really across different regions trying to understand how different factors may be driving these trends, such as climate change. A big finding in this realm typically relates to having fewer freezing degree days in the winter. Warming winters means ticks survive. But there have also been a lot of studies looking into human-induced change to the landscape, such as forest fragmentation and the cascading impacts that may have on deer activity, which has impacts on where ticks are found and how many of them carry disease. But there's not great understanding around how fire, both ignition and suppression activities, are impacting what we're seeing. And so one place that we can start in trying to think about how prescribed fire may influence tick abundance would actually be thinking about the opposite and how mesification, the shift from these open canopied xeric forests to more closed canopied dense mesic forests may relate directly to suitable tick habitat characteristics. So as we have densification of multiple levels in these forests, from the ground all the way up to tree canopy, we have a lot more buffering within these systems to promote a humid microclimate, which is important for tick habitat, as well as vegetation structure that provides the ability for ticks to climb upwards, to seek animal hosts such as deer and small mammals, as well as really dense leaf litter and duff that ticks get to survive in over winter. So these are really important characteristics that fire, as we know, can interrupt. And so first, we wanted to turn to existing studies to see perhaps if we could extrapolate trends elsewhere to this region. Unfortunately, we found no published studies looking at ticks and fire in Pennsylvania. And of the existing studies, they primarily relate to Lone Star ticks, or they have inconclusive results for black-legged ticks. And additionally, we all know that fire management contexts differ greatly across these different regions. And so what we may find in the Southeast with Lone Star ticks may not apply here with black-leggeds. So all of this together informed our two primary research questions. First, we wanted to know how black-legged tick abundance varies among fire-treated and fire-excluded forests, but then we also wanted to further unpack which ecosystem characteristics in these forests may actually influence the trends that we're seeing to get as much information as possible. And some general hypotheses coming back to tick habitat suitability traits associated with mesification. If we reverse that thinking and we consider the time following a burn, we anticipated that shortly following a burn, we would see the lowest number of ticks. Perhaps they're immediately dying in the fire itself, depending on severity and timing. But the conditions created by these burns immediately create a hot and dry microclimate. There's reduced leaf litter due to fuel being burned up, and a more open understory. But then with some time and regeneration, that microclimate starts to bounce back to be a little bit more humid, a little cool and shaded. There's more leaf litter for ticks. And then we expect to find the most amount of ticks in fire-excluded forests, where it's even more stable, it's older, it's had a lot more time to create that nice buffer for the ticks to live in. And so to test these hypotheses, we went right out here in our backyard in Center County. First, we considered sampling across PA, across various different state game lands where the Game Commission is doing burning, but due to resource limitations and what was actually feasible within a summer of sampling, we focused in here. So here's a map showing the 4 game lands we focused in, 103, 100, 60, and 33. And then we set up 24 plots across these game lands, trying to capture a variety of these burn classes that we created. So we have fire excluded plots that served as sort of controls. And then among the burn units that we could feasibly sample in, we separated those into what we considered a medium return plot, which had been 3 to 5 years post-burn, or a short return burn plot, which was 0 to 2 years post-burn. And among these plots, generally, forest types spanned red oak mixed hardwood, dry oak heath, and dry oak mixed hardwood. And then at each one of these plots, we set up 3 100-meter transects where we systematically sampled ticks every 10 meters. And then we also characterized vegetation cover, forest structure, microclimate, which included temperature and humidity, as well as long-term microclimate conditions, which I'll get into in a moment. If you're not familiar with how you estimate tick abundance, one way to do this is through what's called drag cloth sampling, which you see here. Just involves dragging a white cloth across the forest floor and through vegetation, and any ticks that are out questing, looking for an animal host, think it's an animal, and they'll latch onto it, and then you can count the ticks that way. I mentioned that we also classified and characterized microhabitat as an important level of influence here. So while we sampled ticks, we also used a handheld kestrel meter just to measure ground and air temperature and relative humidity. But we also wanted to capture long-term variability in these two characteristics using continuous data loggers. So that's this picture on the left. It's just a little kestrel drop that collected humidity and temperature readings every 15 minutes over the study period, which ran from June to August of 2022. And then additionally, we classified different cover classes because we wanted to get an idea of how vegetation cover and structure may be impacting what we see. And we looked specifically at litter cover, mosses, grass, ferns, and forbs, as well as total vegetation. And then lastly, to try and get an idea of any influences broader forest structure might be having on the system, we estimated tree density at 3 different size classes, as well as canopy cover using a spherical densiometer. And then coming back to our research questions, after we had gathered all of this data, to answer the first question, which was simply just wanting to understand how tick abundance varied across these different burn treatments, we used an ANOVA. But first, we classified our 24 plots into 4 distinct classes so that we could compare both burn versus unburned treatments, as well as time since burn as a potential important variable. And so here you can see we have these short return burn and medium return burn plots. And then we have control plots, which were designated as short return or medium return based on which type of burn they were adjacent to. And then things got a little bit more complicated when we wanted to relate all of these different environmental characteristics to tick abundance. So we ended up converting our 4 burn classes into just presence and absence. So whether or not a plot burned or not was a variable of interest. And then we just ran some stepwise regression and found our top model from there. So jumping into results, I want to preface all of this by saying that everything you hear from now on is only relevant to black-legged nymph ticks, because when we sampled, we didn't find any eggs or larvae, only a couple adults weren't enough. So it's important to contextualize that here, that this is just one phase of the life cycle. But overall, we found about 173 nymph ticks across all plots, with most of them being found in fire excluded or our control plots, and the fewest number of ticks we found at our short return burn plots. And looking a little bit closer at the distribution across all of this data, we found a significant difference across all 4 burn classes, but what I really want to draw attention to is the right side of this plot here, where we're comparing our tick abundance by plot of our short burn and control groups. And we found a 12 times increase in the total number of ticks found at our control plots. And while we did see an overall decrease in ticks found at our medium return burn plots, it wasn't as statistically significant, so we can't draw as strong of a conclusion there. And then when relating different environmental and forest characteristics to these tick abundance trends, we found quite a few potentially important variables, as you see here. Among the variables that negatively impacted tick abundance, fire presence was the most important. It had the strongest influence, followed by several different types of vegetation cover, humidity, ground temperature. And then of the variables that we tested, only a few of them positively impacted tick abundance, and those included grass cover, moss cover, and ground humidity. And so digging a little bit deeper into each of these, based on sort of the level of influence these factors have, first, we found that tick abundance increases with post-fire regeneration time, being the lowest immediately following a fire. And the way that we classified our burn plots, we were able to find that short burn return intervals of 0 to 2 years would maximize tick control, but we're also aware that this may not align with current motivations, objectives, and especially resources for that frequent of a burn interval. But there are a few limitations here in that I mentioned it's only relevant to nymphs. We sampled in the summer and we only sampled one, so it would be good for follow-up studies to sample a few more times, maybe multiple seasons throughout the year to get a better idea. And this really just looks at fire presence and absence, and short return and medium return. We didn't have the ability to account for fire severity and seasonality in this work. Looking at the influence of microhabitat on ticks in these systems, we found some interesting nested timescales of humidity influence, where we found at the time of sampling these one-time static measures of humidity, ticks were found in those plots that had high humidity, which is expected, and also not found in plots that had very hot and high ground temperature, which is makes sense given tick life histories and the cycles. But interestingly, we found from our continuous data loggers that temperature, well, actually ground relative humidity variation was a really important factor, meaning that more variation through time and humidity is bad for ticks. So they require both high humidity and consistent levels. One finding that was a little bit less clear and unexpected is we anticipated more leaf litter to be important for promoting tick habitat, when in fact we found the opposite. But this may just be related to our drag cloth sampling success. If there's too much leaf litter, we might not be able to detect the ticks that are much deeper down. And then interestingly, when looking at our different vegetation classes and those, how they impact nymphs, we found that it was relative on type. So with grass and moss cover, we found that more grass and more moss cover resulted in finding more ticks, which might be related to their ability to sustain suitable humidity levels. Typically, moss requires a lot of moisture, whereas the opposite finding was found with forbs and fern cover, where a lot of forbs and ferns actually resulted in us finding fewer ticks. And we know that this may also be related to our sampling methods. And lastly, we weren't really able to extrapolate any clear findings relating ticks and forest structure. We'd anticipated that tree density would actually positively influence tick abundance, given findings from studies elsewhere. However, this may be related to the fact that in our model, tree density and fern cover were positively interacting, meaning that at plots where we had really high fern cover, we also had really high tree density. So it may be that ferns are overpowering this relationship. And additionally, other measures of forest structure either weren't important or were just excluded because they were too related to the other variables. And generally, this agreed with our hypotheses. We did in fact find that with time following a burn, tick abundance increases. And we also were able to connect this to microclimate conditions as well as vegetation cover. And from all of this, there's some important conclusions to draw. First, we were able to confirm a co-benefit of prescribed fire here being the reduction of black-legged ticks. Specifically, that frequent burns may be most effective, even although this may not align with other stewardship objectives, as I said earlier. And unlike other tick fire studies, we were able to draw relationships between different levels of ecological influence, expanding from microclimate to vegetation, filling important gaps in tick fire research. So now we have a more holistic picture of how ticks and fire may be interacting across landscapes. And importantly, this brings in a really important human health component to prescribed fire that maybe not everyone's always thinking about. If we can restore and manage our forests, both for forest health and human health, everybody benefits. And lastly, there are so many people that aren't noted here that I would love to acknowledge, but I'd like to thank my field techs, Jared Bowman and Ali Holland, and my data assistants, Maya Higgins and Xian, as well as my advisor, Erica Smithwick, and committee, including Alan Taylor and Kim Van Meter, as well as some other really important collaborators on this project. including Jesse Cry, Erica Mochtinger, Jesse Ray Evans, and Mike Gallagher. And additionally, I also want to recognize as a member of Penn State, we're a land grant institution, so I'd like to acknowledge the traditional fire stewards of these lands where this work was conducted, who are the Susquehannock peoples who have been displaced from their homelands. And with that, that is all I have for you. Thank you so much for your time, and I would love to accept questions.